Hi, and welcome to a presentation about FHAST, the Fish Habitat Assessment and Simulation Tool. My name is Peter Dudley. I'm a researcher at the University of California, Santa Cruz and NOAA Fisheries, and this work is sponsored by the U.S. Army Corps of Engineers. I'd like to also thank my collaborators in this, Jesse Black, Stephanie Diaz, Ted Herman, Chris John, and Quan Kim. This is going to be a more full description of the FAST model and how it is developed and what it does. There's a shorter version, about 7 minutes in the same channel, that discusses the philosophy of the model. So the FAST model is about anadromous fish. So here's a representation of an anadromous fish life cycle. You can start anywhere arbitrarily. So let's start with the egg spawning event. So adults spawn eggs on a river. The eggs then incubate. Juveniles come out of those eggs and rear on the river system. Once they get to a certain size, they out-migrate to the ocean. They go to the ocean for some period of time to grow. Once they are mature, they come back up the river and spawn again. And so this model looks at the riverine parts of this life stage, so everything except the ocean phase down here. It's currently developed to have in place juvenile rearing, juvenile outmigration, adult migration. And currently we are developing the salmonid portion of it for spawning and egg incubation, and hoping to develop the sturgeon portion of it for egg incubation and spawning. It's also parameterized currently for spring and winter run Chinooks, California steelhead, and green sturgeon. So those parameters are developed for this model already. Let's begin by talking about the general modeling philosophy first of all. So we'll go into the middle here. So first and foremost, the model is designed in concert with the Army Corps of Engineers and NOAA to be useful to managers. So let's look at what useful to managers means in this context. First of all, the model has a graphical user interface, so there's no command line type typing unless a person wants to become sort of an advanced user and do that. It has what I'm calling usable parameters, meaning that the parameters that dictate how the model behaves are not black box sort of objects like GAMs or forest models. They are mostly just regression parameters you can fit with any statistical program. The inputs are spatial, meaning that managers can simply take stuff off maps or shapefiles and input them into the program. They don't have to change those into indexes or score factors or things like that for certain habitats. It's designed to do A/B comparisons, so show managers how things change if they were to change something in a habitat or do a restoration. And the results are designed to be actionable, so if you care about a certain aspect of it, it's supposed to give you enough information to make a decision about the restoration you want to do. Do you want to do B1 or B2, for example? So more about just the general model framework. It's spatially explicit. So as mentioned previously, you can input stuff directly from GIS layers into it, and then it does its calculations in a spatially explicit manner. This is because habitat restoration and alteration, which is what it's designed to look at, is inherently a spatially explicit phenomenon. It's mechanistic, so it tries to be as mechanistic as possible, looking at the mechanisms that drive this fish behavior throughout the system. This hopefully makes it more robust to modeling scenarios outside of historical data. It has a consistent framework, so between the current fish that are in there, like salmonids and sturgeon, the entire model doesn't change, just some aspects of some calculations and parameters change. These things combine together to make the model generally expandable, meaning as long as the model general framework works for a fish species or a system, you could move it to that species or system provided you got the appropriate data to parameterize the model and get the environmental inputs. Let's spend a little bit more time here looking at the framework. So just a brief overline of the framework here before I dig into it. Effectively, it takes a lot of habitat type information. It then puts that on a spatially explicit grid that the user makes. It then simulates fish behavior or calculates fish behavior over that grid using aspects of fish behavior and physiology. And the outputs of it are effectively resource use of the fish, effectively energy use, and mortality causes of the fish. So looking here at the grid first, the grid's pretty easy to define by the user. The user will use a GIS program to draw a line down the center of the river. Then in the FAST graphical user interface, they will put in two parameters, one being the linear extent the grid goes out over the river, or the longitudinal extent rather, and the other being the resolution that the grid should have on the river. So there's lots of inputs, and we'll go into some more detail about these. These inputs include photo period, habitat, temperature, turbidity of the river, food, available shade, depth and velocity, and predation field. So first of all, let's start off with habitat. So habitat is defined basically by 6 types of things. So there's the 4 substrate categories, which are fine, gravel, cobble, rock, and then the 2 cover categories, which are aquatic or aquatic vegetation, and woody debris or wood. The way these are inputted in the model is through a shapefile. So the user can either find a shapefile for the river, maybe someone has done some habitat classing of it, or make their own shapefile. Fill out the attributes of this shapefile, and then when you put that as an input into the model, it'll automatically be sampled over the grid, and each of these grids will grab the appropriate amount of wood or fine or gravel that habitat needs to have. Let's discuss shade, another spatially explicit thing. So shade is modeled at 3 points throughout the day, and the area over which those shade things cover are taken to be the shade. So it's modeled at basically halfway between sunrise and sunset, and then halfway between the midpoint and sunrise, and halfway between the midpoint and sunset, so 3 times. Then that makes a shade layer which goes out over this grid represented down here, and the grid will then tell you for any one month how much shade each grid cell achieves. So let's zoom in and look at an example of that. So we'll go up here first. So for example, imagine you just had a pole, like a lighthouse or something. This is what the shade algorithm would look like. So the numbers here are the months. So January when the sun is low, and this is up on the Sacramento River area. So in January when the sun is low, we have our 3 sort of times of shade. So that's the shapefile it'll produce to calculate shade over the river. And then as you see, when the sun gets over higher in July, they're far more spread out. And then back into longer shadows in December. What this kind of looks like, this is some habitat shade on a river. So this red section is sort of tree stands that run along a section of a river. This is what it might look like as you go throughout the months of the year, what the shade shapefile will end up looking like. Again, it's tighter in July because of the higher angle of the sun, and longer in the winter months. Final thing we'll talk about here is depth and velocity. So we want to get depth and velocity at every cell point on the river. And what the user is going to input is actually the flow. So they'll have a time series of flow data, which they get from a hydrograph, or perhaps they just are going to use one of our built-in models that can do distributions of flow based on some statistical parameters. But either way, every day there's a different flow for the simulation. And so to interpret or to interpolate the depth and velocity at each cell on the river, what we need to do is have pre-built a series of rasters that show the depth and velocity on this river section for various sets of flows. So here, for example, I have represented a section of the river, and I have a depth and velocity map for 150 cubic meters per second, 200, 250, and 300, just represented here. So the way this works is that you'll have 2 sets of those, one for depth and one for velocity. Every day the model is going to go and look at what the current day is and find its flow. So for here, for example, it'd be 214 cubic meters per second. It's then going to go and interpolate between the 2 closest of those, so 200 and 250 in this case, and get out a 214 cubic meter per second raster. Then every single one of those things is going to be projected onto a cell in the model. So each cell will have a depth and velocity for a given day. Let's discuss energy here for a second. So part of the model being expandable is that we want to have different physiology of different fish be able to be used in the model without excluding other options for other types of physiology. So currently in the model we have green sturgeon, steelhead, and chinook, and as you might know, green sturgeon are a more low energy species than steelhead or chinook typically. So one might expect that their metabolic rates would vary differently with some of the things that we often consider metabolic rates to vary with, like velocity of swim speed, mass, or temperature. So rather than have a single prescribed model that dictates the formulation by which mass, temperature, and velocity will affect metabolic rate, we have a larger model which has various parameters in it that you can turn on and off as constants to either activate or deactivate those things. So for example, all species in here have a relationship which is to log of metabolic rate is a constant times log of mass. But there's two different options for velocity. You can either have a square root of velocity relationship or a just straight up velocity relationship. There's two options for temperature, and there's two options for whether or not you have an interaction with mass and temperature. If that interaction is log mass and temperature or log of both. And so down here is a representation of how that's shut on and off given our current fish in the system. So everything that is blue is turned on, everything that is yellow is turned off. So for example, D1 is turned off for green sturgeon and steelhead, but is turned on for chinook. So that would mean that green sturgeon and steelhead have a relationship that is log temperature. And then chinook has a relationship that is just temperature. And the other things are also true for all those. So you can choose what formulation you want your metabolic relationship to look like depending on the type of fish species you have. All right. So let's go back out and look at some specific aspects of this model. So first of all, let's look at adult migration. So adult migration, what we'll end up getting out, I'll discuss a bit more here about these things, but effectively it's path length and energy cost distributions. So we'll have a section of the river that the fish is going to migrate over. It's going to have a different depth and velocity things for each day. One thing you can do in the model is you can dictate what depth raster you want the fish to be able to migrate through. So if, for example, you only wanted fish to be able to migrate through the various deepest cells, you could say only migrate. The only cells you can access are ones that are available when flow is very low. So sturgeon, for example, migrate during deeper channels than chinook do. So you can restrict their path migration, which is represented here in this red section. So even though the blue river is the wetted section, the red section might be what is wetted during low flows. And so sturgeon, for example, could only migrate through that if you wanted to set the model inputs to be that way. So what this is basically going to do is calculate the optimum path every day that an adult fish could migrate up the river. And it'll do this for every day that you say in the model that adult fish are in the river and give you this distribution over the, weighted by how many fish are in the river over that time the fish are migrating. So to zoom in on what it looks like, effectively we have cells here on the river, and the fish is going to be in one line of cells, and then it's going to make a decision to go straight, go up and to the right, or go up and to the left. And as you might have, if you are a keen observer, you might have noticed that the length between these cells might be different because the curvature of the river might shrink or expand the cells. That's accounted for in the model. So the actual lengths the fish travels here diagonally, straight or up, is actually calculated directly in correct units as opposed to sort of skewed units if there's a high curve in the river. Fish migrate at different heights above the river bottom, so you can dictate in the model how high or low your fish migrates, and that can give it a benefit of effect of the wall velocity on the fish, represented here by the different velocities reductions given heights, given certain free stream velocities. So sturgeon, for example, often are known to migrate lower in the river column than the chinook or steelhead, so they might experience a lower velocity given the free stream velocity you inputted into the model. To do this, when the fish migrate, they also engage in burst swimming and swim above their U-crit, and so we have a very fast algorithm designed to model this based on a more complex algorithm that's very computationally intensive from Martin et al. 2015. So the way this simply works, and I won't go into too much detail here, but it uses a series of linear relationships between these endpoints here to interpolate when a fish should swim below its eucrit, stay at its eucrit, or engage in burst swimming. If it engages in burst swimming, it then needs to recuperate that energy later on in its swim trial. So it is a accurate model description that allows fish to be less efficient swimmers to get through places where it is actually the more efficient method to burst swim and make it out. Some of the parameterization of this is just some detailed information here about Chinook and sturgeon. So eucrit, which is, you saw just in the previous graph, which is the maximum swim speed they can swim at and still be in their aerobic phase, is defined by two things: an inverse relationship and then what's called a beta sigmoid. This looks complicated, but it's actually a pretty easy model to parameterize. It just has two parameters, one being the maximum and then one being, I believe, 25% to the left of the maximum. This shape here allows you to have a asymmetric peak distribution, which is often what we see in things like responses to temperature curves. And so here's just an example of some of the parameterizations we did for steelhead and Chinook. This is temperature and fraction of eucrit they can achieve for that. Here's the same thing with green sturgeon, and because green sturgeon are limited in some of their data sources, we had to use critical thermal maximum to peg the highest temperature. Another example of the critical length, or sorry, the critical swim speed is a function of length, seen here for Chinook, and then seen here for green sturgeon. Again, data limited, so we use some lake sturgeon to fill in the farther out section here. All right, so once you've done this, again, as I briefly mentioned, you'll effectively get some histograms which show you the most common paths the fish will take for migrating. And then you'll also get a distribution showing you the energy costs the fish will take over the time that all these fish have migrated up the river. All right, so now let's go to juvenile rearing. So juvenile rearing, like everything in the model, goes on a daily time step. And juvenile rearing is effectively an interaction between the fish dying from some occurrence and growing to a certain point when it'll either have died or outmigrate from the river. And so let's talk about predation risk first. So there are several ways fish can die in the river from stranding or poor condition, but we'll just zoom in here and talk about predation risk. Predators are distributed in a heterogeneous way on the landscape. So there's not just a constant predator field as in some previous models. So you can see here there's a predator habitat rating, which dictates how likely predators are to be in certain areas. Our current model is parameterized for bass and pike minnow. And then there's a mortality risk, which shows in relationship to a fish of a certain length here. This is run for some length fish the user would input. What's the mortality risk given this predation field and given other things like turbidity and things like that on the river? So let's zoom in and talk about the predator rating and how that gets developed. So predators are placed in a certain zone based on 6 different criteria. So velocity, the type of rock substrate, presence of aquatic submerged vegetation, the depth of the water, presence of woody debris, and shade. And all those are based on just simple logistic relationships that dictate whether or not those things are sometimes there or how present they are. And that gives you a probability of a predator being there. So then once a certain number of predators are placed in the cell, they have what we're classifying as an activity, which is a sort of area in the cell that they occupy. This is used to represent the probability of a prey hitting them. So if a predator occupies 100% of the area of a cell, there's a 100% chance that predator or prey will hit them. If it occupies some fraction, there's some fractional probability that relates to that. That's how likely a prey is to encounter it. The activity goes up with temperature. So this is relativity activity of a predator as a function of temperature. This is parameterized for some predator fish, common as Chinook on the river. Oftentimes in literature, this is thought to be driven by simply your ability to escape predation or do burst swimming. So similar things like that, the differential relationship in predator ability to engage in burst swimming versus prey ability. So then there's also vulnerability we have to consider. So we've established why predators are there. We've established you've had an interaction with them. So how vulnerable are you to that interaction? So that depends on three things: length, cover, and turbidity. So length is used using gap length calculations or gape length calculations for the predator types we have in this river. So there are effectively exponential relationships between those from this paper right here. Cover, you can get cover from vegetation and wood, and then sometimes from sometimes from benthic substrate like cobble and rocks if the user decides to turn that on. If not, then those are left off. You might have realized that what you input in the model is fraction of cover, and what we have that parameterizes the effective cover for saving prey from predators is distance to cover. So we translated that by using this equation, which is simulated by taking a bunch of different fractional random shapes, putting them in a model and simulating the distance with which it takes to get to cover. That gets us, that gets us that relationship right there, which is this regression. And so then we can translate the fraction of cover in a system to the distance to cover. Turbidity also has a very gentle relationship with the ability of a prey to resist predation. That's just based on actual mortality risk of prey, tagged prey going down a system. Okay, just going to zoom out from this. So that is risk from predation. So let's go back out and talk about feeding. We'll start by talking about green sturgeon feeding. So green sturgeon are search feeders. They go along the benthos, vacuuming up prey. So the way we model this is using a random walk. So for example, imagine a fish, and he's going to go over cells. In each cell, he's going to eat all the prey in every one of those cells. And he might backtrack over a cell, in which case he won't eat any prey there because he's presumed to have already taken it all, and on to the next one. So for this, we modeled the cell size to be effectively the width of the fish. The width of the fish is not something the user inputs, or it's not something the program tracks very closely, but we use this assumption to calculate it that effectively the fish has the density of water, which is within a few percentages of what they actually are. So you can get the width of the fish by simply taking the mass divided by the length and square rooting that, assuming you think the fish is basically has a squarish type profile. From that, you can use a random walk calculation to dictate the number of steps a fish will take across a grid which has resolution size W. So the number of steps it's going to take is the swimming speed. If the speed is in body lengths per second, divided by width times length, multiplied by a constant, which changes it from seconds to days. And then, 1 minus p, where p is the photo period because it is assumed that sturgeon feed at night. The user can also change that if they want to. Then we use this equation, which is just if that's the number of steps you have, and the resolution of your cells are W. The unique area covered is just A of F. And so that can end up being how much area you covered, and then we assume that the fish takes all the food in that area. Fish can't feed indefinitely. It hits a Cmax, so Cmax is parameterized using the same sigmoid beta that we saw earlier in the U-crit speed. Again, for sturgeon, we had to use some critical thermal maximum data to peg the uppermost limit of it. Where there is food, benthic food for sturgeon to go is dictated by the user. So the user can simply say it's going to be fine gravel, or maybe there's also benthic food and cobble they want, so they can add that to that. And then otherwise, sections with things not dictated by the user, there's no food there. Let's talk about salmonid feeding. So salmonid feeding is drift feeding. The fish hang out in the water column and catch prey that goes by. I'm going to go back here and talk about this first. So first of all, the intake of the fish is a function of capture success, concentration in the water column, velocity of the water column as it goes by, the area over which the fish scans, and the temperature. So let's look at capture success. Capture success is dictated by the velocity of the water relative to the critical swimming speed, and then critical swimming speed, as you saw earlier, is determined based on an inverse equation multiplied by a beta sigmoid. So then the area over which the fish scans is a function of the reaction distance, which is determined based on the length and a turbidity function. And then the turbidity function is just a function, decaying function, seen here parameterized for steelhead and Chinook, that increases or decreases the probability of capture as turbidity increases. And then the area over which the fish scans is taken to be 2 times this reaction distance d, assuming that d is not deeper than the depth of the water, otherwise the depth of the water, seen here as little d, takes over. So once the fish, sorry, I'll go back out here. Once the fish has done that, number 2 is to make sure it's not below Cmax. Again, Cmax is calculated as this beta sigmoid. Here we can see the data for Chinook salmon and steelhead. Again, asymmetric peak distributions. It then goes into the metabolic rate, as we discussed before, so I won't go back more into that. But once the metabolic rate and the energy intake is taken into account, you get the change in weight, which is effectively the intake prey divided by the energy expenditure of the fish. And if the fish is at a healthy weight, it'll then gain some length, which is defined by this healthy weight function and a relationship between length and a healthy weight, which is parameterized inside the model. And so then the fish will grow. So those 2 things go on, and the fish makes a decision about where to go every day based on risk from predation, stranding, poor condition, high temperature, and its ability to intake food. So let's look a little bit at that. So the fish has effectively 2 modalities of making this decision. 1, it is in good condition. 2, it is starving or bad, poor weight condition. So if it's in good condition, it considers predation and food. If it's in bad condition, it just considers food. The fish is going to choose a habitat cell to go to based on the food there and the predation risk. And it's also, because path selection is spatially explicit in this model, going to choose the best path to get there and potentially suffer predation risk along that path. So if it's going to this green cell right here, it's going to have evaluated these cells, decide the green cell is better than the red cell. And then not only that, but it'll decide that this yellow path here is inferior to this green path here because it suffers lower predation. So it'll move to that green cell here over this green path. So the fish makes that decision every day, day in, day out, until either it is large enough to outmigrate from the system or it dies from some event. Let's discuss fish outmigration very briefly here. So fish outmigration depends on a couple of things, and is effectively the change in velocity the fish experiences over the day. This is to represent high flow pulses cause fish to outmigrate. The photoperiod for some of the fish, and then the length for some of the other fish. So this is just a logistic probability of outmigration, which is the percent difference between a running average velocity and the current average velocity in patches in the max move radius. So did the fish experience a high change in velocity? It can cause them to outmigrate. Photoperiods are parameterized. This is for salmonids, so Chinook and steelhead. And we have 2 in here, one for spring one and one for winter run Chinook. So they're parameterized based on exit data in our system. And then length also affects it. And when the prey decides to outmigrate, just like in habitat selection, they choose a path which minimizes predation risk, but makes them as efficient a migrator as possible, so they cover as much distance while minimizing predation risk. And so those are the current modules in the system. We're actively developing the salmonid spawning and incubation one, as I mentioned earlier, and then hopefully we'll begin developing the green sturgeon one for spawning and incubation. So outputs from this model, I'm just going to give you some brief examples of what they might look like for these actionable results. So for example, here's just 3 charts, and there's a ton of data output from this model. We put them out into 3 separate reports, one for general habitat, one for juveniles, and one for adults. And then there's lots of raw data that a user could put into a GIS program or their own analysis program if they wanted to get more information. But here's just 3 small graphs taken out from those reports. So for example, this is an A-B comparison between a section of the American River where we had some light cover from wood in a certain section, then added in some more wood in different sections. The scales here change between them, so make sure you notice the change in scales. But as you can see here, that the wood made the predation risk less concentrated, so it's more spread out, and then also generally lower. The increased wood had a slight effect on poor condition and also decreased predation. And then you can also see other more detailed things in this that can help explain why these effects are going on, which might be important for managers. So for example, you can monitor a Chinook profile of growth as a function of count of fish in the river and daytime to see if more fish are using habitat to grow or things like that. So that is an overview of the FHAST model. Hopefully it's been informative. If you have any questions, feel free to reach out to me. And thank you for your time.